And for those of you who are starting in spring, like they will register you for the classes. So once you are accepted to the nursing program, you don't really register yourself. They put you in the classes. So that's a little bit different system. Okay, so as we start out, what questions or things do you all have that would be helpful to go over for the lab exam tomorrow?
Like the stations.
The stations, yeah. We'll talk about the stations. I think I want to show Canvas as we talk about the stations. They get my email fixed and all password stuff fixed. Okay, so when you, and I have some things more to say about stations too than I have last week. So one thing to note about stations is that I think there are 11 stations and there are 12 of you all. So when you're rotating around to stations, like, don't crowd up on somebody, just kind of give them some space. And when that station is open, then you can go over to it. And normally the one that gets backed up the most is that camp multi-test plate, because there's only one of it. Because I want everybody to look at the exact same thing and have it exactly right. So that one, people tend to have to wait because it's got 4 things on one plate. But the first station is going to be a microscope with stickers. And it's got A, B, C, D, E, F. And whatever the sticker is on, you write down what is the name of that part of the microscope. Then there'll be 2 microscopes that are already set up with a slide, and they are already in focus and all that. And you look at the slide and say, what is the gram category? Is it gram positive or negative? And that's a 2-point thing. And then what is the cell morphology, the shape? So is it a rod or is it a coccus? Because that's all we've got. It's one of the ones from class. And then that's a point apiece. And then what is the cell arrangement? Is it chains? Is it clusters? Or is it just single cells on their own? And that's 1 point apiece. Then the next station is going to be just a blood auger plate. And it's got a staph strain on it. And the question is, what kind of hemolysis is this? It's next to a window for convenience, so you can hold it up. And you just write down alpha, beta, or gamma for the hemolysis. And then the next one around is to do a catalase test. So you will want gloves on when you first come in. Put gloves on. You don't have to bleach down the benches, but just put gloves on so that when you're going around to stations, you will already have them. And so you'll be at the sink and there's a plate with colonies and there's slides and there's plastic loops and there's peroxide. And you do a catalase test while you're standing there and you look at it and you write down the result and then you drop it in the bleach beaker and you go on to the next thing. Next one is a mannitol salt auger plate, and it'll have a strain on it. And you've got questions about that plate and then about the result and several questions about that. None of y'all are colorblind, right? I didn't ask because you're all girls. It's sexist of me. Girls can be colorblind. It's good to know. If you're colorblind, you can get assistance from me verbally telling you what color it looks, but I think you're not colorblind, so. Then I think the next station is a BIOL esculin slant. A tube on its own, and it will have a result, and there's a question, maybe two questions about that tube. And then there's the CAMP multi-test plate where you got the two discs and the perpendicular stuff, and it's 1 centimeter apart. And that plate, there's 4 things that you can tell from it, and I'll be asking you about all 4 things. So you've got the bacitracin response and the optogen response. I may have those flip-flopped, but the disc says what it is. And then you have the stuff going on here, which tells you, does it make CAMP factor or not? And then the one that people tend to forget is this also tells you the hemolysis of the strep strain. It looks like a locust now. So that one, there's, want to study all 4 things and know what they look like. And on this, this set of pictures are really useful to see what that looks like. And I try to put that one next to the window too. So this is a student named Constant Tolentino. He was a men's volleyball player. This was his plate, and you're going to be looking at the antibiotic response. And this one, those antibiotics did not stop the strain from growing, and you don't really have zones. So they're both resistant. This part is looking at what's the hemolysis of the strain by itself, and it's kind of faint, but it's beta, if you saw it in real life. And then this part, where you're looking at the shape of that junction zone, that's got the arrowhead, so that's a positive. That's CAMP positive. So this one would be optogen resistant and bacitracin resistant and beta hemolysis and CAMP factor positive for those 4 things. Let's look at another one, because I feel like that's the most difficult test to interpret. There's so many things on there. Okay, here's another one. Different strain. This one, there's no zone around this. It's just the bacteria growing how they're growing, and the drug didn't stop what they were doing. So this drug, whichever one that is, it's resistant to it. But this one does have a zone around it where it's blocking the bacteria from growing. So that one would be sensitive. If we look at the hemolysis, what type of hemolysis is that?
Can we close the window? I have like a boat background.
Yeah, there's a glare.
Oh yeah, pull that string. Also, the string blocks the glare a little bit. What do you think?
Would it be gamma?
You got some kind of clearing out there. So it wouldn't be gamma, because gamma is no hemolysis. So then you get down to, is this beta or is this alpha? How do you tell between beta and alpha hemolysis?
That green.
Yeah, alpha has the green. That one's more of a clear. So remember that down here, whatever Staph aureus is doing, that's always going to be beta. So it kind of gives you a comparison. Does this look the same as this? Or does this look greener than this?
Greener.
It looks greener on here, but it's actually beta. Might be easier to see in real life. And then this segment, where these are coming together, it is mostly like that. You don't have a big wedge shaped zone. They're just kind of running into each other and not changing each other's zone. So this would be a camp negative right here. We'll look at all the examples of these. Let's see the next. So in this one, this disc, whichever that one is, it's stopping the bacteria from growing. You've got a big ring around it. This one's sensitive to that drug. It's resistant to that drug. And we've got something going on with the blood agar, but it doesn't look like that. This is the alpha. That's easier to see in person. And what about camp factor? Is that camp factor positive or negative? That's definitely negative. Oh, let's see. I think I had a picture of. I guess I don't have a picture of the one that's negative for everything. So that plate, that's one that people tend to slow down on, and that you need to wait for people to get finished with it. So what's left after that is the gram-negative tests. So for gram-negative tests, you'll have 2 tubes and a plate to look at for gram-negative tests. There we go. So there will be a McConkey plate, and it will say on the plate MAC, so you know what plate it is. It won't have the strain names on the plate. What they will say instead is number 10 for like station 10 or something like that. And so you look at the McConkey plate, and there's either going to be colonies that are clear and see-through and not purple, or there'll be colonies that are purple and not see-through. And so these are positive for lactose fermentation. These are negative. So there'll be that plate and 2-3 questions about what's going on that plate. Oops, do the next here. Then there will be a urea agar tube, and it will either be yellow or pink or kind of in between, like a weak positive or a total negative. And so the question will be like, what can you tell about this strain based on this test result? And maybe one other question about how this agar and how this color system works. And that's usually station 10 or something like that. And then you'll have a sim tube that's already finished. So it'll either be black or not. And it will have the drops on top of it already. So the drops might be red or the drops might be clear. And that tube also has several questions about it, because you can tell different things off of that tube. And that's station 11. Then the other thing that is kind of a station is for you to, I kind of sit where the chalkboard is. And when you get ready to come out and pronounce your strain, we go out in the hall and I'll point to one on your written down list and I'll say, this one. And you say it and I circle the numbers like 3 points, 2 points. And you go on and do the rest of your test. Or some people do that. Some people want to do that first and some people want to do that last.
So do we pick the strings?
No, you write them down.
Okay.
So you have a space to write down the 11 strains and tell what's their category and hopefully get them spelled right. And then you bring your list to me and I point to one. Yeah. And if I think that you have it so messed up that it will mess up your pronunciation, I won't point at that one. Mhm. Yeah. You can do it. Just take your time. I think remembering the 11 strains is one focus. And then the second one is looking over stuff from your notes about how these tests work. I might have some papers to give back to you. Does anybody else feel like I have your papers to give back or notes or anything like that? I think I've given most of them back, so. And this will be tomorrow at 2. So I'll open up the door at 2 and you can come and get started. And if you feel like people are all crowded up on the stations, just sit down and wait a while. And chill and relax yourself. Actually, you won't be here. Caitlin won't be here. She has COVID. Just going to tell you. She'll have to take it later too. So yeah, that'll be tomorrow. So today we're going to start on kind of a new unit, but it's continuing to talk about immune system. And we're going to be in this slide set for two different days, both today and Wednesday, talking about specific immunity or acquired immunity. There we go.
This is unit 4.
Yeah. Yeah. Unit 4. So specific immunity can also be called adaptive. And it's operating in a very different way than innate immunity or acquired immunity works. And today we're going to talk about what makes, what are the characteristics of specific adaptive immunity and how it works to produce antibodies, like my doll here, or like this cartoon, which is a different way to show an antibody, and how those antibodies protect us from shapes called antigens. So the antibody is one of our best protective proteins in the immune system, and they have a Y shape, kind of like this. And antibodies bind onto specific shapes called antigens. And I think we talked a little bit about those shapes in Unit 3 when we talked about PAMPs, or pathogen associated molecular pattern. And PAMPs are shapes also, but PAMPs are shapes that lots of pathogens have in common. So let's first look at what are the characteristics of specific immunity. And this is a list to memorize, kind of like you memorized the characteristics of innate immunity. So the first characteristic is that for you to activate this response is kind of slow. So it's not always ready for you to use it. You have to develop it. So it's slow to develop, and it takes about 2 weeks after your first exposure to a pathogen. And this response is tailored or is adapted to a specific 3D antigen shape. And normally that specific antigen shape is something that's not that common amongst all bacteria or amongst all viruses, but something that's really specific and unique to a certain pathogen. This immune response requires learning. So we're going to have certain immune cells that have to learn to recognize a specific shape and do some tasks to get ready to provide us immunity against it. This is our third line of defense. We don't need to use this unless pathogens or microbes get past the barriers, which are the first line, and also get past those processes like fever and inflammation and phagocytosis and so on. So if they get past those, we use these defenses, and they're slow, they are adapted, or they're specific, and it requires our immune cells to learn. Once they do learn, this is a strong and long-lasting defense. And it's long-lasting because this style of immunity develops what we call immunological memory. Our immune system learns a particular shape, and it remembers how to fight it for a lot of years if this is working well. And then finally, this branch of immunity is going to use B cells, T cells, and antibodies for the defenses. B cells, T cells, antibodies. B cells and T cells look kind of boring, and they do not make good dolls like this, but the antibody looks interesting, and it makes a nice stuffed animal doll. So this is a list to memorize. And as we go through, we kind of talk about how does it do all this stuff? And why is it slow? So right at the beginning of this unit, we have the graph for this unit, and this is one of the most important, probably the most important graph in the field of immunology or learning about the immune system. And it's showing how much antibody does a person make over time. Not just any, let me rephrase this, how many specific antibodies a person is making against a specific 3D shape or antigen. This is concentration of antibody. And across the bottom, we're looking at time. So time has gone by. And we start this process with what we call initial exposure. And if you would circle this on this graph, the initial exposure begins when our body senses a new foreign 3D shape. That's the initial exposure. And from the time that we sense that, our acquired immune system will start to make antibodies, and it takes a while. It's kind of slow. Then we build up eventually a protective level of antibodies after a while. And that first slow buildup, that's called the primary immune response. So we get this really slow buildup of antibodies, and it takes a while, but we make them. And then the antibodies will kind of decline until later on we have the secondary exposure. Okay, so you got the initial exposure, or some books would call that the primary exposure. And you make some antibodies slowly, and that's pretty good. And then some amount of time passes, and then you're exposed to that same thing again. So our body is going to see or encounter the same exact 3D shaped antigen. And the second time that we see it, we get a faster, stronger buildup, really quick. And that's called the secondary immune response. It's kind of like when you learn any new thing, like the first time you see it, I don't know what's going on. It's taken me a while to understand it. Slowly, I kind of get it. Then maybe I sort of forget because I didn't look at that in a while. But then when you go back and review that same stuff, oh yeah, I got that. I remember that. Like if you had to go back and review from AMP1, you already had AMP1. How does muscle contraction work? Well, then, okay. And then second time, oh yeah, much quicker, much stronger remembering the second time around. So that's called secondary immune response. And when we say that people have true immunity to something, what we mean by that is that they can already do a secondary response. So it's not their first rodeo with this antigen. it's not their first time making antibodies against that thing, and the second time around, they can really fight it off well with antibodies. That's the secondary response. And that first word is true. True immunity. Like I should be immune to getting chicken pox because I got exposed to it in the second grade in April and I got sick from it. And then in May, I had to miss all the exciting end of the year second grade things because I was sick. But then after that, when I would get exposed to chicken pox, I made good antibodies and I fight it off. But I can't go back and go to the Girl Scout first ever camp out or field day where what was I gonna do? Like, I think I was gonna throw something. Whatever kids throw at the field day. I had to miss it. Because I didn't yet have a good immune response because I just encountered chicken pox and I was trying to fight it off with my slow, weak antibody response. So this graph, we come back to it at the end of this slide set and look at look at it again with new knowledge. So when our body is looking for shapes to determine whether they're dangerous or whether they're not, every molecule in the world has shapes to it. At a microscopic molecular level, they have shapes. And our immune system, when we're young, it learns what are the shapes that belong to us, and we call that self. Like we're made out of shapes. And when we're young, our immune system learns what's part of us. And then after that, anything that is non-self is considered foreign. And for our immune system, what is considered foreign is considered dangerous. So it's like if, like with kids growing up, you know, whatever they're used to seeing does not seem dangerous, but if they see something they never seen before, they might be afraid of it, think that it's dangerous. So for our bodies, new shapes or new antigens, our immune system will consider them dangerous. And if you think about a particular pathogen, like let's say E. coli. E. coli is a big bacterial cell and it has shapes all over its surface. And each shape on E. coli, on its surface, is a separate antigen. And the shapes individually, we can call those epitopes. So an antigen can be a substance that has a lot of different shapes, but each individual one can be an epitope. I'm mostly going to call the shapes antigens for simplicity's sake. So when our body experiences new shapes that are dangerous, we'll go through a process of making antibodies. In this picture, the antibodies are shown with this Y shape here. And each different antibody type is going to bind onto a different shape. So we see here, they're coloring them different colors. So the green antibodies are specific for the rectangle shape. And so we would call this antibody type number 1. But we can also make antibodies that bind onto the circle shape, and that would be antibody type number 2. We can also make antibodies to protect us against the dangerous pink pyramid. It's never seen it. It's so dangerous. That would be antibody type 3. So this particular antibody binds onto this particular antigen shape, and then they have kind of a shape match. And the only thing that's not perfect about this doll is that they really ought to come with one more of these, because antibodies can bind usually 2 of the same antigen shape. And that's helpful because that lets them kind of go 2 on 1 and bind 2 things at a time instead of just 1, but it has to be the same shape on both sides. So the way that I think about antibodies is that antibodies are proteins that can bind to foreign shapes and help destroy those shapes plus whatever they're attached to. So antibodies are just proteins that we make, and they are grabbers of shapes or binders of foreign shapes. And when they bind onto these shapes, that's going to lead to destruction of that foreign dangerous thing. That's how our immune system protects us from outside world, is to figure out what's new and attack it with antibodies, basically. So whatever this shape is, it's about to die. Maybe that's why it looks angry. Shouldn't last long if it's bound by an antibody. It will destroy whatever is attached to it. So if this antibody was bound onto this shape, and this shape was also bound onto the owl camera, this would get destroyed as well as the owl camera. Kind of whatever is stuck onto it, touching it. So antibodies, they're proteins, and they have a lot of different parts to them, and they are normally drawn in a Y shape, or as a doll sewn into a Y shape. So this is 3 different ways of looking at them. This is kind of what they look like for real in the protein structure. So they've got this larger piece down here, and then they've got these arms, kind of like this, and the arms are bendy. And they actually bend. They're not like this. And they can flex and kind of bind on to whatever is the dangerous thing. Sometimes they're shown in this type of cartoon. And when you see the Y shape, the antigen part, antigen binding part is out here on the hands or the arm region. And whatever binds here would be the same thing that binds here. The rest of that, don't worry about for this class, what the parts are called, but they generally have a Y shape and they're going to bind onto the antigen. The important part is where does the antibody bind to the dangerous foreign shapes? And that would be on this one, the green parts. And on these parts. That's what binds to the dangerous foreign shape. The rest of the antibody has a different job to kind of get things to happen for the destruction. But the shape binding, the antigen binding, it always will bind at least two of it, and it has to be the same thing twice, like two markers. Or that's the only thing I have that's the same. Two markers. Maybe two erasers. So next what we're going to talk about is, okay, so you combine some dangerous foreign shapes. How is that even helpful? How does just binding protect us from anything? The first way is by binding onto a shape and neutralizing what that antigen shape is supposed to be doing that's harmful. So one example is if you have an antibody bind on to the virus out here, what part of the virus is this? Those are spikes. And what are the spikes supposed to do for the virus? Bind to the host cell. So if virus spikes have antibodies stuck on them, the spikes now can't bind the host cell. And if you have enough antibodies to bind onto the spikes, it's called a neutralizing level, and the virus could be in you and just floating around, but it can't actually infect you. That's a way that antibodies can be helpful. What would you call this thing coming out of the bacterial cell in the red? What is that thing?
Flagellum.
Flagellum, yeah. And what's it for? Helps the cell move around. So if you got antibodies hanging on there, this cannot help the cell move very well. So if you have antibodies on the flagellum, bacterium can't swim very well. And it's not destroyed yet, but it is definitely slowed down from infecting you. And in this case, this is not even alive. This is a toxin chemical, but if antibodies are bound onto it, the toxin can't do harm. It can't harm the human cell or the neuron or whatever it normally would be harming if it's got an antibody stuck onto there. So that's called neutralization. Just by binding on, the pathogen or the toxin is stopped from doing harm. So that's neutralizing is kind of stop the harm by antibodies binding on. The second thing that antibodies could do that is helpful is called agglutination, which is clumping. Agglutination is helpful clumping. And agglutination is possible because antibodies can bind at least two of the same thing at once. And I always think about this, think about a lot of things in cell biology is like toddlers, how toddlers act. And say if they're, you got the two little brothers getting in trouble, or they're getting in trouble and doing something they should not, I could go get them and grab both of them at the same time, one in each hand. Stop it. Which I have done. And I have clumped them together and also kept them from doing what they wanted to be doing because I can grab two at once. And notice here, these antibodies that they're showing would be like 5 of this all together. It's a, what is that, a quintuplet antibody that's 5 in a ring with 10 hands, 10 grabbers instead of 2. So they're grabbing onto bacteria and the bacteria cannot move very well because they're all clumped up with these antibodies grabbing them. Another way that antibodies can be helpful is to bind onto the surface of something and form an antibody coating. So they can coat a pathogen with a layer of antibodies. And that is going to attract macrophages. To eat it. So opsonization means to make tasty. So some people, if you put hot sauce on a food, it doesn't matter what it is, they're probably likely to eat it. Like, oh, is that spicy? I'm gonna eat it. Or with a little kid, if it's got icing on it, they're more likely to eat it. And if a pathogen has antibodies all over the surface of it, macrophages are able to find it and eat it faster. So it's like how people stick a thing on a car and be like, wash me. It's like, eat me, eat me, eat me, eat me. Quicker to come and eat that danger and do phagocytosis. So that's a way antibodies are helpful also. They can agglutinate, they can opsonize, and they can do neutralization. This slide has 2 separate things and the pictures go with 2 separate things. So first additional thing that antibodies can do is they can make these complement holes that we talked about in innate immunity. They make them form faster and more. So you get these pores, but if antibodies are involved with that process, that happens faster and stronger. So it's called complement activation. And antibodies can also make natural killer cells work stronger and faster. So if I were to summarize this, I could say antibodies help the complement system and natural killer cells work faster and stronger. those things would eventually happen anyway, but if you have antibodies, they will, happen more effectively. So we've got a picture here now of, here's a natural killer cell. And in this case, it's not killing a human cell, it's killing a bacterial cell, but the concept is the same. And when it binds onto antibodies, it releases chemicals that are trying to kill that cell. So the antibodies are kind of bringing those two together so that the chemicals are right there killing the cell and protecting us from this pathogen here. Yeah, this will be the slide I wanted to get to for today. So this is a good one to end on. So we make antibodies and all of them have this as their basic shape, but we make 5 classes of antibodies. And antibodies are also known as immunoglobulins. These words mean the same things. So immunoglobulins. And this part means that they have something to do with the immune system, and this part means that they are a small protein. So together, they're called immunoglobulins. And immunoglobulins get this abbreviation, big I, little g, or IG. Immunoglobulin. Humans make 5 different kinds of antibodies, or 5 different kinds of immunoglobulins. And there's different ways that you can remember which versions they are. So you notice they're called IgG, IgM, IgA, IgD, IgE. So together they spell MAG, or maybe they spell gamed. You kind of make a word out of it. I kind of prefer gamed because it's close to being in the order that they, how concentrated they are in the body. It is basically the order of percent that they occur in the body. So I usually use gamed. But I'm going to talk about the 5 different kinds and what's special about them. So the first kind is IgG, and IgG has the usual Y shape, and it is the most common in our blood plasma. So this row right here, where it's saying the percentage of the total antibody in serum, that's just the blood plasma. And if you check a person's blood for antibodies, 80% of those are going to be the IgG type. The other thing that I want you to know about IgG is that it is produced some in the primary response. So we make a good amount of it in the primary response, but it is produced really a lot in the secondary response. You mostly find it in blood plasma. Second one that I'm gonna talk about is IgA. Notice IgA is two individual antibodies that are joined together and by their feet. If we think about them like a person standing like this, it's two together. So there's actually 4 antigen binding sites instead of 2. But the main thing to remember about IgA is that this is called secretory antibody because it's found in secretions of the body. So liquids that a person makes on purpose that aren't urine, those are secretions. So it is in mucus, in tears, in saliva, and in breast milk. This one's super important for new babies. If they're breastfeeding, they get a lot of antibodies from the mom through breast milk, and it's secretory antibody. It's the second most common overall. And you do find it in mucus, but you find it in these other places also. Then we come to IgM. And this one, I remember the M is like for mega. This is the one that has, it's a ring of 5. And they look like a snowflake. It's not a fragile snowflake, it's like a super badass antibody snowflake that could bind 10 antigens at a time. IgM. We call that a pentamer. So it's 5 in 1. 5 antibodies. And they all would bind the same thing. Like really, I should get 5 of these. So awesome. Maybe someday. Get 5 of them. I'm like, woo, we can bind 10 of the same thing. It would all be this. It wouldn't be this and a remote. It would all be 10 of the same thing. And IgM is produced a lot in the primary response. So when we first start making antibodies for the first time, we tend to put a lot of energy into making this type because they become really like powerful, where they've got 10 binding sites because there's 5 together. So we make these a lot in the primary response. And we make some in the secondary response, and they do float around in the blood plasma, available to bind 10 of the same thing. Next one is IgE. We'll save that for next time. I don't want to rush through that. IgE, and then it's Wednesday. We'll be in the same slide set. On Wednesday. Maybe we won't have mowing on Wednesday.
Thank you.
See you tomorrow.